Hello everyone, my name is Jake Sumner and I will be presenting our results in section 4 group 3 for the Zimmerome project. Our goal in this project was to elucidate any protein structural changes that might have occurred due to the mutations in Carl's genome on chromosome 21. Firstly, we decided to choose our genes of interest by selecting those that were protein coding and had the highest mutational burden. Our hope was that we would be able to find missense protein coding mutations using this as our filtering criteria. As it turns out, only 5 of the 10 protein coding genes had any mutations at all, while only 3 of the 10 had missense mutations. Once we identified our proteins of interest with missense mutations, we then ran the wild type and mutated protein sequences through a deep learning transformer network called NetSurfP. NetSurfP approximates the amino acid relative solvent accessibility, or how exposed or buried an amino acid is, as well as the likely state of order of that residue. Residues that are highly disordered are likely to have high relative solvent accessibility. It should be noted that we tried to find PDB structures that modeled residues in which mutations occurred, but we only found one gene where this happened, batch 1, which we show modeled in the slide on the right. Our results show that the G471A mutation in batch 1, the graph on the left, has little effect on the solvent accessibility of the amino acid residues at and around the site of the mutation. You can also see from the ascending red line that the disorder for this region of the protein is predicted to be high. This means that a change in the RSASA likely would have little effect on the function of batch 1, which is canonically a transcriptional regulator. Similarly, we found 3 mutations quite close to one another near the C-terminal tail of GRIC1, an ionotropic glutamate receptor. These mutations, all leucine to serine, have a marked effect of increasing the average solvent accessibility of approximately 40 amino acids in this protein region, which in turn increases the disorder of the region. However, because this region is disordered and none of the mutations are pathogenically linked, we would not expect these mutations to be harmful. Also of note is that APP, or the amyloid precursor protein, the protein precursor to the amyloid beta plaques in Alzheimer's disease, was highly mutated, but none of the mutations occurred in the protein sequence. All good news for Carl.